We are starting with our lessons on prokaryotes. I've said that I'm going to look at the types of cells one after the other. So we talked about eukaryotes, and now we are going to talk about prokaryotes. And for eukaryotes, it was clear that they are the cells or cell-like particles that do not have nucleus. And for the case of viruses, I made you understand that they do not have the full genetic material that would cause them to initiate and control activities of the cell, and thus they do not undergo life activities by themselves. But prokaryotes have the full genetic materials that make them carry out life processes by themselves. But the difference is that their genetic materials are not membrane bound, so they are not surrounded by membrane, in simple terms, that is to say. So for the objectives, at the end of this lesson, the student would be able to name the main groups of prokaryotes, and would as well be able to describe the structure of a typical prokaryote, and would also be able to state the types of bacteria. But this lesson is going to take into account the lesson on types of cells. So if you haven't watched our video on types of cells, please do so before you proceed with us. So let's start by talking about what prokaryotes are. Simply put, a prokaryote is an organism or cell that has genetic material but no nuclear membrane. So that means no membrane-bound nucleus. And some would say that they do not have true nucleus. And prokaryotes as well lack membrane-bound organelles. Now it is important for you to know that prokaryotes are in two main groups. So we have bacteria, then we have archaea. But just like in the case of the eukaryotes where they were exemplified by viruses, in this case too, they would be exemplified by bacteria. So it means the focus would be on bacteria. It doesn't mean bacteria are the only prokaryotes. Right. So we'll talk about the structure of bacteria and then their types. So let's start with the structure of bacteria. I have this image to let us appreciate the generalized structure of bacteria. So many parts have been shown in here, but I'm not going to talk about all of them. So the ones that I'll be highlighting will be what I'll be talking about. So starting with chromosomal DNA, this is where the genes are found. And this is the center for the control of the life activities of the cell. Right. So this is what actually makes the prokaryotes true cells, because they have full genetic materials that are able to control and to say make them carry out life processes. Then there is cytoplasm, which contains all the cell organelles and inclusions. There is an extra DNA that is called plasmid, and I'll be talking about plasmid in one of our lessons later. So for now, let's just keep in mind that plasmid is an extra DNA found within the bacterium. Bacterium cell or the cytoplasm is surrounded by plasma membrane, so that controls the movement of substances into and out of the cytoplasm. And there is cell wall that protects the cell and also keeps the cell in a state of turgidity. Finally, I'll be talking about flagella, and in singular sense is flagellum. Flagella are the structures that are used for movement. The main structures that are used for movement, let me say so. And the bacterium cell would lash the flagella against the water in order to cause movement. So these are the parts that I would like to concentrate on for now. You can read about the other parts later. Okay. So next we are going to talk about the types of bacteria. And for the types of bacteria, we are going to talk about it in two different contexts. In one way, we would classify or group bacteria according to their shape. And in another way, we will group them according to whether they have an outer membrane or not and the thickness of a substance called peptidoglycan. So these are the two main ways we are going to group bacteria. So let's start by looking at grouping bacteria according to their shape. In here we have one type Cocci or Cocci, as some people would put it, and the singular word is Coccus. These are the bacteria that are spherical in shape. Then we have Bacilli or Bacillus for those that are rod-shaped. Then there are Vibrios that are curved rod-shaped. And the Spirilla, singular Spirillum, they are the spiral-shaped ones. And then we have the Spirochaetes, the helical-shaped ones. And I have some images here to help us appreciate their shapes. This is showing only 3 forms. I have another image that will show all of them. So here we see the spherical forms. So there's a coccus, and then we see the rod-shaped ones, Bacillus here, and then we find some spirochaetes here, helical shape. Okay, let me take this one away and let you better appreciate them here. This is a video, so you can pause and then look at them well. So the Cocci here, Bacilli here, this represents the Vibrios, the Spirillum here, Spirochaetes here. I know that some books would group the shapes of bacteria into 6, some even into 4. So it depends on where you are reading from. Now let's move to talk about the other way to group bacteria. So bacteria can also be grouped according to the presence of an outer membrane and the thickness of peptidoglycan. So when you're talking about peptidoglycan or the thickness of peptidoglycan, we mean the thickness of the cell wall, because the peptidoglycan is what makes the cell wall. So here we have 2 types. One of them is gram-positive bacteria. And gram-positive bacteria, they have thick peptidoglycan, and there is no outer lipid membrane. The other type would be the gram-negative bacteria. They have thin peptidoglycan, and there is an outer lipid membrane present in them. And let's look at this image to better appreciate what I'm talking about. This is gram-positive bacteria, and here there is a thick peptidoglycan, and so no outer membrane. This is an inner membrane, so the membrane around the cytoplasm, but there is no outer membrane here because the cell wall is thick enough because of the thickness of the peptidoglycan. And when you compare it to the gram-negative, the peptidoglycan here is thin, so that means it has thin cell wall, and that is also in another way supported by the presence of an outer membrane. So there is an inner membrane to surround the cytoplasm, and there is an outer membrane outside of the peptidoglycan. So this is how we distinguish between them as gram-positive and gram-negative. Okay, so that is what we have for our lesson today, and let's refresh our minds on what we have done so far. In this class, we have looked at prokaryotes and their main groups. We have also looked at the structure of prokaryotes, and then we finalized it with the types of bacteria. Okay, so just like we do, I have these few questions that I would like you to try, and you can send your answers in the comment section or to the contact that I'll be adding. So you have to state the shape of each of the following types of bacteria: Vibrios, Bacilli, Spirochaetes, and Cocci. Very simple. So as we have begun the journey of prokaryotes, next we are going to look at how prokaryotes carry out life processes, and we are going to look at it in the context of how bacteria carry out their life processes. So let's meet in our next lesson.